The Hardy-Weinberg law is the unifying principle in population genetics. It allows scientists to predict how alleles will be transferred from one generation to the next. In order for Hardy-Weinberg equilibrium to be useful in predicting whether or not the alleles in a population are in stable proportions, certain assumptions must be made. Organisms reproduce sexually. Mating is random. Population size is very large. Migration in or out is negligible. Mutation does not occur. Natural selection does not act on the alleles under consideration. The Hardy-Weinberg equation stipulates that the proportions or frequencies of alleles can be restated as two variables, P and Q. Since the sum total of all the proportions of anything must be 1, we can write that P plus Q is equal to 1, where P is the frequency or proportion of dominant alleles, and Q is the frequency or proportion of recessive alleles. If we return for a few moments to the principles of Mendelian genetics, and analyze a heterozygous cross, a so-called monohybrid cross, we can see that in this, the smallest possible population, the allelic frequencies are very straightforward. Since there are two capital A alleles, or two dominant alleles, out of a total of 4, the frequency is 0.5. So, P is equal to 0.5, and Q is also equal to 0.5, since P plus Q must equal 1. If we complete this cross in the normal way, but including the allele frequencies, we get the following results. They can be tabulated in a Punnett square, as you can see on your right. The expectation is that 1/4 of the offspring will be homozygous dominant, 1/2 of the offspring will be heterozygous, and 1/4 of the offspring will be expected to be homozygous recessive, which can be explained mathematically by squaring the Hardy-Weinberg equation. And you get a result of P squared plus 2PQ plus Q squared equals 1. Let's recap. P is the frequency or proportion of dominant alleles. Q is the frequency or proportion of recessive alleles. P plus Q must equal 1. The equation is squared as a result of mating, or that two probabilities mix. So we get the expression P plus Q squared equals 1 squared. And this results in the Hardy-Weinberg equilibrium expression or equation, which is P squared plus 2PQ plus Q squared is equal to 1.
Okay, let's break the Hardy-Weinberg equation down and compare it with the kind of cross that we've done before using the Punnett square. P squared represents the frequencies of the dominant alleles multiplied with each other. So they represent AA in a hybrid or monohybrid cross. (2PQ), the expected frequency of heterozygous individuals in the Hardy-Weinberg population, are then represented in the Punnett square as the two A, small a combinations, or the two heterozygotes. Q squared represents then the homozygous recessive individuals that are expected given the allele frequencies that we have. In our box, or in our Punnett square, we notice that the proportion or frequency of recessive alleles is 0.5 times 0.5 for the other possibility gives an expectation in this small population of 0.25 or 1/4, just as we saw in the previous slide. Let's consider an example and work it through in a step-by-step manner with explanations. A population of flowers was observed to have 500 individuals with 2 distinct phenotypes. Assuming that the phenotypes are the result of one gene with 2 allelic forms, and that one allele is completely dominant over the other, we can begin to formalizing the information about the population. The gray flowered plants are found to express the recessive trait, phenotype, and therefore we can say they are homozygous recessive. Gray equals little a, little a. This means that the other, yellow phenotype, must be composed of heterozygous and homozygous dominant individuals. So yellow flowers are capital A, little a, or capital A, capital A. If the population consists of 150 gray flowered plants and 350 yellow flowered plants, we can calculate the abundance of each phenotype. Let's focus on the 150 homozygous recessive plants. If there are 150 gray homozygous recessive individuals out of a total population of 500 individuals, then the relative phenotypic frequency of the gray phenotype can be calculated. 150 divided by 500 equals 0.3. 30% of the population is homozygous recessive, gray flowered. Let's use the Hardy-Weinberg equation to find the allele frequency for the recessive allele little a. The gray flowered plants have a phenotypic frequency of 0.3. So the genotypic frequency for the homozygous recessive is also 0.3, since the recessive phenotype is caused by the recessive genotype. If the homozygous recessive genotypic frequency is 0.3, then we can apply the Hardy-Weinberg equation to find out what the allelic frequencies for both the dominant allele, capital A, and the recessive allele, A. If the frequency of homozygous recessive individuals in the population is 0.3, and using the Hardy-Weinberg equation, we can say that Q squared is equal to 0.3. Looking at the Punnett square, we can see that 0.3 then belongs in the position for the homozygous recessive individuals. Since Q squared equals 0.3, we can say that Q will be equal to the square root of 0.3. This will give us the allelic frequency for the small a, or recessive allele. So taking the square root of 0.3 gives us Q equals to 0.548. We can now solve for P. Let's use Q to solve for P. Since P plus Q must equal 1, 1 minus Q must equal P. To find P then, we take 1 minus 0.548 and get 0.452. So P is equal to 0.452. Using the Hardy-Weinberg equation, let's find all the expected genotypic ratios assuming Hardy-Weinberg equilibrium. P equals 0.452 and Q equals 0.548. Applying these frequencies to the Hardy-Weinberg equation, we then get 0.542 squared plus 2 times 0.452 times 0.548 plus 0.548 squared, all equal to 1. Having done the math, each of the 3 phenotypes that are expected in the population can now be realized. You can also compare them with the Punnett square to see how they are aligned in the traditional manner. Those highlighted in blue represent the homozygous dominant. Those highlighted in red represent the heterozygous. Those highlighted in yellow represent the homozygous recessive. Once we know the genotypic ratios, or the expected proportions of each type of genotype in the population, we can find the expected number of individuals that carry each type of genotype. Finding the expected number of individuals in the population that have each genotype is achieved by multiplying the frequency with the number of individuals in the population, in this case 500, which gives then 102 individuals that are homozygous dominant, 247 individuals that are heterozygous, and 150 individuals that are homozygous recessive. These are of course just expectations. No real population will have exactly these numbers.